preserving fossils. Alright, bats, birds, and insects, they're not related. Their common ancestry goes way, way, way back. And those are examples of analogous structures because they're produced from, none of those. They're produced from morphological convergence. So 5, none of the above apply there. Okay, all of these describe why the fossil record is biased. So a change in the body form of a common ancestor, that would be morphological divergence. Pangaea was a landmass about 260 million years ago. Small scale changes in allele frequencies describes microevolution. Human birth weight is an example of stabilizing selection because it doesn't really change. If both ends are selected for and the middle is selected against intermediate, then that's disruptive selection. If male and females look different, then that's a term of sexual dimorphism. Okay, the founder effect is a form of bottlenecking. So you have a small group that kind of bounds an area, and then so the gene pool isn't very big there. Okay, evolutionary adaptation improves the odds for surviving and reproducing. Gene flow is the movement of alleles into and out of a population. Natural selection acts directly on phenotypes and indirectly on genotypes. Balanced polymorphism is the term for the fact that you can have alleles, both of those alleles in the population. Change over time occurs if it occurs in heritable traits that can be passed down. Where are we here? Carbon-14 is the most common for aging fossils. If allele frequencies shift in one direction, that's called directional selection. The peppered moth is an example of that. All of these determine the phylogenetic relationship among species. And a clade is a group of organisms that shares characters, a set of characters. Homeotic genes are involved in developmental patterns and sequences. If a mutation has very little influence, it's a neutral mutation. Kingdom is the most inclusive taxonomic category. The protist would be here, the kingdom that is comprised of eukaryotes. 3 domains of life, everything but Protista. Fungi are included in the eukarya domain. The cyanobacteria, which aren't really bacteria, are thought to be the first aerobic organisms. The cyclic pathway probably evolved first. It doesn't produce any oxygen. So the cyclic pathway produces no oxygen. Okay, Earth did contain a lot of water vapor, but no oxygen gas. So 2 of the above is correct on that. RNA probably gave rise to the first living cells. Mitochondria and chloroplasts were once prokaryotes. All of these are good evidence of endosymbiosis. 900 million years ago, most of the major lineages had shown up on Earth by then. Earth is estimated to be about 4.6 billion years old. The stromatolites are the dome-shaped fossils. Okay, for genetic equilibrium to occur, this was kind of easy because you're like, it's either this or this. And so for genetic equilibrium, mating must be random, Bacterial resistance to antibiotics is an example of microevolution. All of these are forms of pre-zygotic isolation. Okay, if there does have to be fertilization, but embryo doesn't survive or is sterile, that's a form of post-zygotic isolation. Physical barriers is a type of geographic isolation, and it's called allopatric speciation. So both A and B are true for 43. This would be lethal. That's answers in the question there. It's easy. It's not a trick question. Genetic equilibrium is never observed in natural populations, so that'd be false. The peppered moth situation is directional selection. Human genome does contain homeotic genes, of course. As an example of a homeotic gene is the Hox gene. It might suppress the DLX gene, and that prevents the expression of appendages. Taxonomy is involved in identifying, naming, and classifying species. And the prokaryotic domains include the bacteria and the archaea.